thank you all for joining our second podcast series entitled Educating the Researcher in Study Design, specifically sample collection and handling. My name is Dr. Candace Ulmer, and I am the Chemistry Branch Chief of the Eastern Laboratory at the USDA FSIS here in Athens, Georgia in the U.S., and I'll serve as today's moderator. Our emphasis is comprehensive study of metabolism as it pertains to a biological system in response to a stimulus or a pathophysiological condition. Both targeted and untargeted metabolomics incorporate workflows with steps that include bioanalysis, data processing, compound identification, and biological interpretation. As many of these steps remain unharmonized and unstandardized across the metabolomics field, it becomes extremely important for researchers to exercise a high level of critical thinking and judgment to ensure the execution of a successful metabolomic study. The Education and Training Committee of the Metabolomics Society has created this podcast series, which will include expert panelists who will discuss the challenges, the strengths and weaknesses of different approaches, as well as decision-making experiences and best practices for each aspect of the metabolomics workflow. Well, when we talk about a successful metabolomic study, we need to ensure careful consideration of the study design. As you can imagine, there are several key concepts that should be considered for each one of those various aspects of the metabolomics workflow. So our inaugural session of this podcast series covered study design from a planning perspective. We covered discussion points associated with sample size, minimizing bias, appropriate controls, types of study designs, as well as the appropriate number of replicates. Well, in today's podcast, we want to explore the second stage of study design, so sample collection and handling. We're going to ask our expert panelists today to share their recommendations and best practices to optimize sample collection and integrity for metabolomics. We will focus on human and mammalian biofluids and tissues. However, we still welcome any questions in regards to sample collection and integrity in our chat. We will ask all attendees to remain muted throughout today's podcast and instead use the chat feature. Feel free to assign any of these particular questions to our panelists in the chat. I'll now like to ask each of our expert panelists for today's webinar, which will be mentioned in speaking order, to provide a 1 to 2 minute introduction. It'll be very helpful if the four of you could include your years of experience in the metabolomics field, as well as your areas of expertise that are relevant to the metabolomic study design. I'll first start with Dr. Jerzy Adamski. Then I'll move on to Dr. Julia Kuligowski, as well as Albert Koulman and Dajana Vuckovic. I'll first start off with Dr. Jerzy Adamski from Helmholtz-Zentrum.
Hi everybody, I'm Jerzy Adamski from Helmholtz-Zentrum München in Germany. I'm biochemist and endocrinologist by training. My expertise and experiences in metabolomics are over 15 years. So my biggest achievements, as I judge them, are in the development of different protocols for matrices, different tissue types, and as well I contributed a lot to sample stability protocols and as well through the study design in large human cohorts. This would be my introduction. Thank you.
Thank you. We'll now pass it on to Dr. Julia, if you could introduce yourself.
Hello, my name is Julia Kuligowski. I work at the Health Research Institute La Fe in Valencia, in Spain. I've been trained as an analytical chemist and completing my PhD at the Department of Analytical Chemistry at the University of Valencia in 2011. Then afterwards, since 2012, I moved to the neonatal research group here at the Health Research Institute, where I also got first in touch with the field of metabolomics. Our study focuses mainly on preterm infants and neonatal hypoxia-ischemia, and we've been involved in exploratory studies and the discovery of biomarkers in this field. We've been mainly employing LCMS and GCMS as analytical techniques, but also vibrational spectroscopy. And more recently, we started to conduct nutrition studies and work with human milk, including the analysis of human milk oligosaccharides as well as extracellular vesicles.
All right, thank you so much, Dr. Julian. We're now going to move on to Dr. Albert.
Hello, I'm Albert Koulman. I'm at the University of Cambridge at the Institute of Metabolic Sciences. I started in metabolomics in 2003, so almost 20 years ago. First in plants, and since 2007, almost exclusively in humans, as well as some work in model organisms. And over the last years, one of our research interests is the development of tools to measure metabolites and lipids in remotely collected samples, so either through dry blood spots or other technologies.
Thank you so much. And then last, we would like to introduce Dr. Dajana.
Good morning, everyone. My name is Dr. Dajana Vuckovic, and I'm an associate professor at Concordia University in Montreal in Canada. So same as Julia, I'm an analytical chemist by training, and I've been working in the field of metabolomics for about 15 years. My research group focuses on developing new analytical LC-MS-based methods for primarily unstable and low abundance metabolites. And this includes looking at new sampling and extraction approaches. In terms of applications, we focus primarily on cardiovascular health, inflammation, and nutritional metabolomics. And in terms of the types of specimens we routinely deal with, this includes all the biofluids as well as different tissues, both from human and mammalian systems. I also have expertise in low volume sample handling, and particularly in vivo solid phase microextraction and its use for metabolomics. What's really exciting about this technology is that no blood or tissue needs to be taken out. So I think this is kind of an unconventional way to do metabolomics analysis, and I'm super excited to be here today as part of the panel.
Thank you so much. So as you've seen, we have a wealth of knowledge here for today's podcast. So given you all's expertise, I do want to ask this somewhat broad question to each one of our panelists. And we'll start first with Dr. Jerzy. So when we talk about this sample preparation workflow, sample collection, sample integrity, what are some key considerations that a researcher should keep in mind when they're beginning to plan their study in regards to sample collection and integrity? I'll first start with you, Dr. Jerzy.
Yeah, so I would say that the first important thing is to have standardized description how to process and collect and store the sample and keep it running. If you grow the cohort, or if you grow with your experiment and change the process of collection and storage, this will be a disaster, because this is spoiling the whole study design, and the stability of metabolites will be showing too much variability. So keep the same protocol. The protocols are available everywhere, and depends on the metrics you're studying. This will be different protocol, but they are already in the web, so freely available.
Thank you. We'll definitely follow back up with you, Dr. Jerzy, to see where we can find some of those freely available resources that you're referring to for some of our listeners. But I do want to transition over to Dr. Julia. You know, when you are planning your study, what are some key considerations that you think of when you are thinking about collection and the sample integrity?
So I would agree very much with what was already said. We need standard operation procedures for sample collection to make sure that all samples are collected in the same way. Also, I would really insist in that all people that are involved in the process need to be trained to provide consistency in the study. And I know that especially when we work with clinical samples, sometimes we have to make a trade-off between what would be ideal for our sample and what is realistic in the clinical setting. So sometimes, yeah, we cannot store the sample immediately, but at least we should make sure that we establish, for example, a maximum time frame that is admissible, or take notes of deviations from the protocol.
Thanks so much for that information, Dr. Julia. One key aspect that you mentioned there was, you know, things that are specific to that clinical setting. So we absolutely will follow up with you a little bit later on in our podcast about specific clinical guidelines and some information in there that's very pertinent to sample storage collection. So thank you so much. Dr. Albert, what about you? Are there some key considerations that you keep in mind when you're thinking about collection and stability?
It's very dependent on the scale and the type of study. So if it's small, then the person who will be running the samples can also be involved in collecting the samples, but we've also been involved in very large-scale projects, and there it's really crucial the training of people collecting the sample. That's where probably the biggest unwanted variation can come in, is if the people collecting the samples aren't properly trained or trained in a different way. So I think for people involved in the study and analysis, it's really helpful to be aware how people are trained or be involved in the training to make sure that the people who are collecting the samples are aware of what they can, what the impact is of their handling on the final result.
Absolutely, Dr. Albert, I completely agree. Generating those SOPs and ensuring that all laboratories are following them, I think, is absolutely critical during the integrity of those samples. And then lastly, I just want to follow up with Dr. Dajana. Do you have any additional collection or sample integrity key points that you consider when planning your experimental design?
Yeah, of course, I agree with everything that the other panelists have mentioned. I will add to this to worry about, lots of materials that are being used for sample collection, and to use the same lots, especially if it's a multi-site study. This may not be done during planning stages, so this is really important. And then the other question to ask yourself is there some particular pathway you are targeting? And if yes, are the analytes from that pathway known to be unstable? So do you need to introduce some extra controls in your design, or will the standardized protocol work for you?
That is so true. Thank you so much, Dajana. So Dr. Jerzy, I just want to follow up really quickly on something that you mentioned. You talked about the necessity to have, a consistent workflow or a consistent protocol for each of the samples that are involved in a study. Could you talk a little bit about, for some of our beginners in the metabolomics field, how do you even begin to create some of these protocols? Should we be optimizing our workflows and our methodologies for the given matrix or the analytes of interest that we're interested in analyzing?
This is definitely the case online now, that you have to adapt your standard operating procedures to the type of study you're performing. If you are doing any experiments with cell culture, it could be done in this way. They use snap frozen your material very, very fast. If it's an animal model, the processing will be similar to that of collecting the RNA analyzing. So quickly and snap frozen in liquid nitrogen and then passed to storage at -80 on liquid nitrogen. If it's a plasma or urine or serum, the protocols will be different. The most prone protocol for artifacts is preparation of serum, because this allows the clothing period could be done overnight, it could be done in room temperature, it could be done in cold room or whatsoever. So many people tend to have it described, but the people who are doing that, they modify it. So it's maybe wise to have a short description how the individual sample was actually in a clinical setting prepared. If it was 1 hour or half an hour or 2 hours in any temperature, so that you can see if this projected into the creation of outliers. So this is for serum. For the preparation of human urine collections, if it's spontaneous urine, it's the best to freeze it. After that, if it's a 24 hours urine, it does not have to be processed, because you cannot freeze the aliquots. You just collect the 24-hour urine and that's it. It's pretty much standardized. Some analytes, like oxylipids, will require addition of, for example, organic solvents, 50% final concentration of methanol, for example, will stabilize prostaglandins and so on. But you cannot do it in a clinical setting. So this will be a subset. If you are covering a specific pathway, the SOP has to be designed to keep this metabolite stable for this specific pathway.
Mm-hmm. So what I'm basically hearing from you is that our researchers need to be considering not the matrix, whether it's going to be, serum, plasma, but all tissue, but also we need to be considering what analytes we're interested in measuring, whether they be specific lipid classes, some pretty volatile metabolite species, so forth and so on, at the very beginning stages of our study design. And I think that is absolutely crucial. So thank you so much for sharing that. Dr. Albert, you had mentioned in your quick explanation something about SOPs. Where do we even find those SOPs? Is the laboratory creating them? Is there some reference online where we can find them? Can we grab them from another university and adopt them in our own lab? How do we even start with the SOPs and ensure that all of our laboratory personnel follow those?
Well, there's a few questions in there, so I have to be careful on how I answer them all. So yeah, there's a whole range of SOPs available online, and I don't think that anybody would object that their SOP is used by other labs. That's why you publish them. So that's why you make them available because if you make them available, you're probably convinced that they're a good SOP, so you want other people to use them. So that's one part. Making sure that people stick to them, that's a whole different question, and that's something that you have to think very carefully about, especially in larger studies. And probably the larger your study is, the more careful you need to think about the simplicity of your SOP. So if you have a very complex SOP and you have lots of people involved in following the study, there's a very high chance that something goes wrong. So if you have lots of people involved in the study, you probably want to keep it as simple as possible. So we do, for instance, also studies that people collect their own blood sample at home. And that's the extreme version of it, where you have to make sure that everything is as simple as possible so that the chance of people doing something wrong is minimized. So I don't think you can blame people in your study for doing something wrong. If something goes wrong, your SOP was either not clear or you've made a too complicated SOP.
Thank you for that information. I wanted to follow up with you, Dr. Julia, about the clinical aspect of this. So as mentioned by Dr. Albert, there are SOPs that do exist, both in published literature as well as online, and can be shared in laboratories. But how does that change for the clinical setting? Are there also SOPs? Are there specific clinical guidelines that can be followed? How would this need to be tailored for the clinical setting?
So I would agree very much with what was already said here, and so we need to make our protocol fit for purpose. It's important to include all these spots during the design of the study. So we will probably have to do some prior tests to see, for example, if the metabolites that we want to measure are stable in the conditions that we use. And it's very important to have, especially in the clinics, to have simple protocols. Otherwise, it will not be possible to obtain samples that you can afterwards use for interpretation. Especially also when we think about home collection. And to add on what Albert said, I also think it's very important to remind people of the collection of the samples when they have to do it at home. So in our protocols, we usually include for our staff a task that covers to call the participants for home collection prior to the date of collection, so that they don't forget about it.
That's very key. Thank you, Dr. Julia. So, Dr. Dajana, I just wanted to follow up a little bit on what Julia just mentioned here about these guidelines and these SOPs. As was, kind of implied by some of our panelists, if there are deviations from that SOP, if there are mistakes that are being made, oftentimes that means that we need to clarify some SOP to make it be a little bit more apparent. But even still following an SOP, there may be areas where we introduce some contamination. What are some possible areas where we can introduce contamination in our workflows when we're talking about sample collection?
Like I was saying before, all the reagents, storage devices, your tubes, cryovials, pipette tips that you're using to aliquot your samples, so all of these can introduce contamination in the samples. The other thing that you have to worry about, especially if you're working with either low volume samples or with matrices that are very aqueous, such as urine, is non-specific adsorption. So for some of very hydrophobic compounds in urine, we see a lot of loss with the walls of the container. So if you don't account for that in the design, you don't accurately measure the compounds in urine. So that's definitely something to worry about as well. And then in terms of contamination, the rest of the process, sample preparation all the way to LCMS analysis, we can introduce contamination along the way. So having blanks that cover the whole process really helps to know the sources of those contaminations.
That's very key, important. And what I'm getting to is that a lot of the areas of contamination could often too be very specific to whatever instrumentation that you're including, what specific matrix you're including within your overall sample design. So I want to dig a little bit deeper and talk about some of these matrices here, as well as the volumes and the size of our study. Let's first talk about the matrices and talk about sample collection. I'm going to mention urine first. Does it matter where the urine is collected, whether it be at a clinic or at home, when we talk about sample collection? I'm going to target this question to Julia. Are there specific considerations when we talk about urine collection and where it's being collected?
Yes, definitely there are. So it's very important if urine, for example, is collected at home, that we provide all the materials that have to be used, also with regard to the contaminations that have been mentioned, and that we explain all the procedures very well to the participants. That includes also storage and important also the transport, because typically the samples are then transported from home to the lab by the participants. We also need to think about, to instruct people about the conditions, how, when and how they have to collect samples. For example, if we need fasting samples, we should think here of the study group, but also especially of the control groups, because we typically encounter more problems regarding this in the control groups than the study groups. Yeah, and as I already said, if we want to collect urine at home, you will need to remind participants in order to avoid the loss of samples.
That's perfect. Thank you so much. So Dajana, just to follow up on this, when we talk about urine, how do we deal with urine collection maybe in younger cohorts, like children, or are there specific considerations that we should be aware of for urine collection in those sample groups?
So maybe I'll turn over this question to Julia, because she's more expert in pediatric sample collection. So I will, we've done it more for mammalian model studies, but Julia, maybe you can add a little bit on working with children.
Yes, so in the case of blood samples, we are very limited in the volumes when we are working with newborns. Also, we have to think of if we collect more invasive samples like blood, then probably less people will be willing to participate in our study. So we usually prefer to work with urine. And here, basically, when we work with babies, we have two options. Either we can use adhesive plastic bags. They have inconvenience that there's the adhesive that sticks to the skin, and that might cause skin irritations. So typically we try to be even less invasive and use cotton pads that we place in the diapers and then express with the syringe. That also allows to collect the volume of sample that is needed. We just need to repeat the process in order to increase the amount of samples.
Thank you so much for that information. I want to open it up to our other panelists. Are there any other key considerations that are maybe specific to the urine matrix that we should keep in mind for collection?
Yes, if I may, it was said already that we need home ability to collect the urine samples. The blood collection is still limited, but it's possible. What I would like to address here is the time over collection. So it has to be recorded. If it's set to be fasted, it's okay. Most people will comply to that, but the time of collection, because of the circadian rhythm, will be showing completely different appearance because of glucocorticoids, melatonin levels, and so on. Metabolism is changing during our circadian rhythm, so it has to be noted when the sample was collected, whether it's blood or urine.
I would like to add something to it as well, especially if you ask people to collect samples at home, either urine or blood samples, but let's focus on urine, is to make it very clear what you want, but also make it clear that if something changes, it's not a problem, but they need to write it down or make you aware that something is different. Because I've seen studies and have been involved in studies where we've been too strict, and then people don't want to commit that they've made a mistake, and then you will find it out in some of the samples, but you don't know where the problem is. So it's much better to make sure that participants are aware that mistakes are made, and that it isn't a problem as long as they record it and that you know where what to expect from the samples.
Yeah, I think that's key, and not even just specific to urine, but across multiple matrices. So transitioning from the urine matrix to now talking about blood, more specifically dried blood spots, because we did get quite a few preceded questions about these dried blood spots, how to handle them, how to collect them, what type of specific tools we can use to get more of a consistent spot. I want to target some of these questions to you, Albert. When we talk about dried blood spot sampling and the collection of these dried blood spots, could you just kind of give us a quick overview of what that looks like? Are there certain devices that work better than others? Are they fairly stable?
So I think the most important thought you need to have when you're working with dry blood spot is that they're the worst type of sample that you can collect. Especially if you ask people to collect their own sample at home, there's lots of variation. It's very difficult to get a consistent sample, and just spotting blood onto paper will lead to a whole range of artifacts in the sample collection. So enzymes will remain stable, and your glucose will change, and all your antioxidants will change. So you need to be aware that working with a dry blood spot will introduce lots of artifacts in your metabolite profile. So you have to be very careful when you analyze the sample, that what you analyze and how you can interpret that. So I think that's, if you're aware of that principal problem that there is with a dry blood spot, and you take that into account, only then you can use safely a dry blood spot for metabolomics and or for lipid profiling. So you first need to be very careful and understand what you're doing. Then there are a whole range of different devices now available to either collect blood samples at home or to spot blood onto paper. I think it's really dependent on the analytes of interest. So which part of the metabolome are you most interested in, as well as the scale and the type of study you're doing, that you really need to think about, what device can work best for me. A simple finger prick and spotting onto paper is by far the cheapest option available, and will give you the opportunity to look at quite a big range of metabolites already. But there are lots of other technologies available that you could use that can give you either a specific sample type or a specific volume of sample, and that can all be advantage for certain research questions. But I don't think there is a one-size-fits-all answer for it.
Yeah, no, I appreciate your insight for sure. And Dajana, I just wanted to help have you just follow up a little bit on some of this information. I know that you have some experience working with low volumes, and just kind of moving away from the dry blood spot matrix to maybe more of that liquid sample that's very, very low volume. Are there any key considerations that we should keep in mind when we're working with low volumes for sample collection and ensuring sample integrity?
Yes, so when working with low volumes, this issue of non-specific adsorption becomes even more prominent. So you really need to do those tests and make sure that your analytes are okay. And then the other issue that you often have when you switch to very low volumes is there is more demand on your analytical method. So you actually need to optimize your analytical methods to be able to get the coverage you want from this small volume sample. So on my side of things, we always optimize methods. If we are going to do very low sample volume collection, it necessarily means some development into optimizing the analytical part of the method as well.
So for some of our beginners that are on the call, when you talk about like optimizing the method and making sure that it's specific for the analytes of interest, could you elaborate a little bit more on like what you mean? So what are some steps that we should take in quote unquote optimizing our method?
So when we optimize our LCMS methods, we look at things that will boost sensitivity when working with low volumes. So things that can ionize, that can enhance ionization. So for example, do you need to add a derivatization step if you are working with very low abundance metabolites, for example, something like estradiol and those pathways, or oxylipins and those pathways. Separation as well, so LC separation to avoid having suppression, because that will also help enhance your signal. So I think those would be the two main areas. And then, can you pre-concentrate at all your sample? Usually when you're working with very small volumes, you don't have that luxury, but if you can do any kind of pre-concentration, so this is where techniques like microextraction come in, where you can then inject everything that was extracted directly onto the instrument after the sample cleanup with microextraction.
Gotcha. Thank you so much for that information. So Jerzy, Dajana was mentioning a good bit about having to tailor the protocol to account for some of these low volumes, and that may mean in certain instances adding some additional steps in your workflow, which of course may affect the overall stability of some analytes. So are there like certain metabolites that you would consider to be more stable than others? And where can we maybe find a list of some of those more stable and less stable metabolites?
Actually, I wanted to add something to the low volume. So there is one aspect which is maybe obvious to people who work with that, but it could be as well difficult to notice it when you are working below 50 microliters. Okay, so if you have 50 microliters in your Eppendorf tube and it's frozen, so the moment you're taking it out of the freezer, probably there is some evaporation or sublimation taking place in the tube. And you don't have 50 microliters at the bottom of the tube, but it's all over the tube. So you have to first thaw the tube, spin it down, and handle it. Otherwise, you're losing a lot of metrics by that. So this is something which is overlooked. The second step, when taking out 10 microliters for processing, for lipidomics, for example, you don't need much more. Consider evaporation process and how much is outside your pipette and how much is inside the tip of your pipette. So where is this 10 microliters you're handling? So this maybe it's not that difficult to observe when you are doing it by hand with multi-channel pipette. But if you are switching to robots, you're handling hundreds of samples, then the 10 micrometers tend to be much less at the end of the process in comparison to the beginning because of evaporation. Sorry for mixing this slow, but I thought this is important to add into it. So,
absolutely. I'm you gave us this additional information. I'm sure our listeners on the call are very grateful as well. But yeah, those are absolutely some key considerations for low volumes that we should take into mind when we're designing our experiment. And, you know, even with those low volume considerations, like I mentioned before, we still have to be very considerate of the stability of some of these analytes of interest. And as we continue to add additional steps, we need to be even more and more mindful of the stability of some of our analytes as they go through these additional steps in the workflow. So can you quickly talk, Jerzy, about stability, sample stability, and things we need to be considerate of? Are there certain metabolites that can serve as like a marker for stability?
Yeah, so this is very important, and the information about that, how stable are metabolites, it's not stored, to my knowledge, not stored in one place. You can have a look at different common data places like Human Metabolome Database. I'm contributing to that all the time, but it's simply not systematic. But some of the rule is if you are dealing with metabolites which are mediators of signal transduction pathways, they are usually very unstable. So, for example, 2-hydroxy vitamin D derivates, which are signaling, not D3 and not D2, they are short-lived. Yeah, prostaglandins, 2-alpha, prostaglandin 2-alpha, in human blood is less than 1 minute at a normal pH, but metabolites of them, PGM2M, for example, is very stable. So this is a very common description. Now, some processes like deamination, oxidation, they are taking place everywhere, and they are very fast. So if you are doing with research with polyamides or amino acids, lipids, you have to know that the low concentrated molecules will be disappearing faster. And highly concentrated molecules maybe are more stable, but they are not changing very much in the under normal conditions. So I would focus rather on these pathways where the low concentrated metabolites are showing the changes. And there are several indicators which are in public domain, like deamination, like oxidation, like lipids being processed to different classes, which give you overall description how was the overall stability of the sample. So you can check it. But for very given metabolites, the information is rather within the biochemistry books and not in the metabolomics knowledge.
Can I
That's really
add one
yeah,
part
I was
to that?
ready to throw it.
So lipases in blood samples. You have to be very careful. For instance, if you're looking at lysophospholipids, because a lot of lysophospholipids can actually be an artifact of lipases from your sample collection. So that's something you need to be very careful with.
So Albert, to follow up on that, are there any additives or things that we can add to our sample, like antioxidants, BHT, et cetera, to kind of help with the stability of some of those lipid species?
So I've always been very hesitant to add things to a sample to make it stable, because that makes it really difficult to get it uniform and reproducible. So if you have a targeted method, for instance, we do a lot of work on just vitamin C analysis, and there we add MPA to plasma samples to stabilize it, but then we use that sample only for vitamin C analysis and not for something else. So so if you know what you're measuring, then it can be a useful tool. But if you're more interested in general metabolite profiling approaches, I would be very hesitant to add something to it. So,
Julian, like in the clinical setting, when we're collecting samples, are they collecting certain types of tubes, or are they stored certain ways to ensure sample stability? Like with your
So actually, yeah. So as Albert said, usually we do not add any additives, because if we want to use it for a global method, we are like, these additives are always optimized for a certain class or type of compounds, but not for all metabolites, of course. So unless you really want to look into certain metabolite classes, like for example oxylipins or glutathione, then we would not add anything. So what we typically use for storage of our samples are dried sample tubes, like Eppendorf tubes, sometimes we use opaque tubes if we work with, or if we might want to look at metabolites that are not stable with light, UV radiation. And important, yeah, we try to process them very quickly so that we can store them at the earliest time point possible.
Dajana, I know that you have a lot of experience with some of these unstable metabolites. Could you just, for some of our listeners who are kind of unfamiliar with more stable and less stable metabolites, give us a couple examples of some of those unstable metabolites that we need to be considerate of in our workflows?
Yeah, so for example, oxylipins or the eicosanoid pathway, there is a lot of unstable metabolites there, and also lots of possible artifacts due to oxidation of endogenous oxylipins and precursors. So this pathway is kind of fraught with both stability issues and interferences. So that, I think that would be a good example. And then Jerzy gave some other examples of vitamin D and in general the signaling metabolites. I'll add there, if you're working in animal studies, one way of looking at these unstable metabolites is with something like microextraction, because there we don't co-extract enzymes. So right away during sampling, you do metabolism quenching step. Then the main pathways you have to worry about after the sampling is, oxidation and other non-enzymatic pathways. And we'll be doing some studies very recently where we are showing the devices can protect also from oxidation, all of these oxylipins. So there is some newer techniques that can help you deal with some of these very unstable signaling molecules.
Oh, that's very helpful. Thank you, Dajana. Albert, I follow up really quickly because I know that you have some expertise working with like some of those larger studies. So let's say that, the design was that you're going to have multiple freeze-thaw cycles, and what are some key considerations that our listeners should take into account when they do have these larger cohort studies that are involving multiple freeze-thaw samples or freeze-thaw cycles, in regards to collection and stability of the analyte?
Yeah, so it's a bit of a black box what's really happening during freeze-fall cycles, and it's also dependent on the sample type. For instance, dry blood spots are really bad samples for freeze-fall cycles because you get condensation during the thawing that can bring water into your sample and get enzymes active again. So you have to be really careful, especially with dry blood spots, how you store them, that they stay dry, and then if you thaw them, that you make sure that they don't become wet again, but stay dry during the thawing time. And with other, if you take just say a plasma sample or a serum sample, it really depends on what you want to measure and how much the freeze thawing will affect it. Yeah, so we prefer to work with samples that didn't go through a freeze thaw cycle. If you end up with samples that have been through freeze thaw cycles, then it's usually that the running a metabolomics or a lipid profiling analysis is an afterthought of the experiment and not part of the original design. So yeah, it depends on your research question, but you need to take it into account that these samples haven't been handled in the best possible way for doing metabolic profiling. So yeah, it will automatically be a suboptimal approach.
So when you design your larger studies, then how do you try to account for not having to have multiple freeze-thaw cycles? Like, what are you doing in your design to ensure you're not going to have to do multiple freeze-thaws?
Yeah, so preferably we want specific aliquots designed already, so that there are aliquots that are stored in the right format. Especially on a larger scale, that's really crucial as well, because you want to be able to use the sample container in the pipeline of your metabolic profiling system. So that can either be that the fit into a 96 well format or something like that. So so in that way, if you're doing larger scale studies, thinking about that design and which sample goes into where is really crucial at the start.
Absolutely. Yeah, I think ideally you want to account for that as much as you can in the initial.
Yeah.
I want to open any of our panelists at this point to kind of ask any additional tidbits about stability or to talk about freeze-thaw, those controversial freeze-thaw cycles.
Julia, you wanted to say something?
Yeah. Actually, it was already said somehow, but I really would like to make a statement here that you should invest, or like an advice, you should invest some thought, or like a lot of thought actually, during the planning phase of your study to avoid that you have to use samples that underwent several freeze-thaw cycles. So we really plan our study ahead and think of all the methods that we would like to use or to apply to our samples so that we have a specific aliquot for each of them.
Mm-hmm. Absolutely. Dajana, do you know of like any indicator metabolites that might be helpful to assess like multiple freeze thaws and like, you know, the integrity of your sample?
So I know some groups, I think Marcus Wenck reported sphingosine phosphate as one of those metabolites. I think in general, this is an area where there is a lot of active research. So I would say we generally check lysolipids, as well as Albert was mentioning when we are running our untargeted method. So I think there's a little bit more work that needs to be done on really biomarkers of stability. But yeah, sphingosine 1-phosphate was proposed as one. I usually like to look at glutathione ratios, things like that as well.
Thank you so much. So we focused a good bit on liquid, dry blood spots, you know, more of our blood-based assays. What about tissue? Are there specific considerations that we should keep in mind, like when we're collecting tissues from mammalian studies? I'll target that over to Jerzy, since you have a lot of experience with a lot of different matrices.
Yes, tissues are more complicated because they are actually all contaminated. Okay, so if you are dealing with a liver sample, an animal study, it's contaminated by blood. If you are dealing with blood, it's contaminated by signals from all other tissues. So you have to consider it. Some people try to perform metabolomics on perfused liver or perfused kidney. It didn't help much because there was a lot of artifacts caused by the perfusion. They have not improved the clearness of the picture, what is actually specific for the kidney or liver and what is to be seen in the plasma or serum. So actually the best way is to subtract the metabolomes of plasma or serum from that of the tissue and look at specific signals. Usually the processing of the tissue is actually less prone to artifacts due to the instability of a metabolite. Somehow the tissue environment is protecting stabilizing the metabolites. As soon as you do it fast and in the same way and snap freeze the tissue, it will stay very well reflecting the status at the moment of collection. What is very much contributing to clearness of the signal for a given tissue is how well defined it is. So is it contaminated by other tissue? If you are collecting gonads, the usual contamination will be fat. If you are collecting some parts of the brain, you have to consider that must be a very good collector and people doing preparation to dissect specific region of the brain. I mean, cerebellum is very, very easy, but what about hypothalamus and all small nuclei there? It's simply, yeah, it's a state of the art. It's really demanding. So these could be contaminations, which I think are reacting, progressing into the signals which you are seeing in the tissue. Sometimes the lavage, lung lavage, so putting fluid into the lung and collecting it, it's very good matrix for the analysis of functionality of epithelium of the lung. it happened to be very much reflecting the situation in icosanoids and other molecules. And it was a big surprise how quickly these molecules could be released to the environmental fluids, which are usually not present in the lung. So yes, the collection of tissue is actually less complicated if you do it in a defined way, and if you apply the tissue preparatory techniques for that.
Oh, wow, that's amazing. Thank you so much. Are there any other panelists that want to add maybe to the collection of tissues and some key considerations here?
Sure, I'll chime in. I'll actually disagree a little bit here with the panelists. I'll say tissue can be more tricky if you activate the pathways, right? Or if you're working in animals or clinical situations where you have the effects of anesthesia, drugs administered, antibiotics, things like that, because then your metabolic profile reflects all of those things. So you have lots of confounding factors in addition to what you're trying to study. And I think for oxylipins, I mean, this was shown a lot in brain studies that if the brain is collected and dissected using regular procedures, you can have up to like 500 time release in prostaglandins and other oxylipins. So the only way to stop that is to do like head focused microwave irradiation prior tissue collection. So I think for some pathways, we don't have as much stability as we would like. So you have to worry what pathways you want to target and how to best collect that tissue sample. This is where in vivo types of approaches work well, because then you don't have anesthesia. You're working with, awake, moving animals, so you're getting more of the real metabolome in that sense.
We see that contamination term rear ugly head once again in our discussion for today. To our other two panelists, are there any additional comments that you want to add about tissue collection?
Well, it really depends. If it's a human tissue, then yeah, we often see that the use of anesthetics can have a big effect. Certainly, you see, a whole range of new compounds that you normally don't see in a human sample. Then that's just the what's used to alleviate the pain. And with animals, yeah, it's what needs to get some thought in is the type of tissue and how to homogenize it, and what's the best way of doing that. But usually those are small scale studies, so that you have some room to think about it and work with it. So
I have to speak. So, sorry, this is scientific discussion. So yes, anesthesia and tissue collection. So if you do this error, that you have a mistake, that you have a control animals killed by the CO2 or something like that, and the test group under anesthesia, because you have to collect it under the beating heart, something like that. These are confounded. You are collecting completely different groups. And the use, as you said, Dajana, the use of different anesthesia in the same type of animal, it will cause completely different metabolism. This is clear cut. This has been published, and I took it, I have forgotten it. Thank you very much. So we don't disagree. This is a very major factor. Sorry for interrupting.
Diane, did you want to add anything, or
no? I think we are
last. All right, sounds good. So in our last couple minutes, I do want to talk about some of the portions when we talk about collection. So Julie, I'm going to target this question to you. In the clinical setting, are there some alternative approaches to more of some of those invasive sample collection techniques that you use?
Yeah, well, so as I said before, we typically try to work with non-invasive samples, like, for example, urine. Urine is probably the most widely employed matrix for us in the clinical studies. It also has some inconvenience, of course. For example, if you want to relate it to a clinical event or some kind of event, then there might be a time lag, so you will probably need to study or get an idea first on how on when to collect the urine sample. Also, you cannot specifically select the time point of sampling usually, especially when you work with babies or children. Yeah, so you will have to wait for the sample to be collected. Blood samples, it's much easier to establish a fixed time point. We try to work, when we work with blood samples, we try to use small volumes, even dried blood spots for some purposes. Well, yeah.
So even with you collecting your little bit, that's not invasive anymore, but are there ethical considerations that you should be mindful of even when collecting urine in a clinical setting?
Yes, you have to be careful, of course, on how often you collect the sample. You have to explain it to people beforehand so that they can decide whether they agree or not and sign the informed consent form, of course. It has to be very well established when the study starts, when the study ends, how many samples will be collected, how much volume of each sample will be collected, and how all the procedures will like all the participation of the patient need to be established very clearly beforehand. Yeah.
So in other words, patient consent pretty much through every process within the workflow in the clinical setting?
Not necessarily, but they have to be informed about each step so that they know what they are agreeing to.
Mm-hmm. Yeah, thank you for clarifying that. I do want to open it up to the rest of our panelists to see if they have any additional tips or considerations in regards to the ethics portion of collection.
Yes, if I may start. So indeed, this is changing very much in last years. There was a new regulation which is implemented in Europe. At present, we cannot use any sample from human study without patient consent. The patient consent has to include the information, what is the purpose of the study, what will happen with the samples after the study was completed, destroyed, and so on, and how the data will be used in the scientific process. Not exactly description of all possible procedures, but describing, is it a metabolomics, metabolite analysis, is it a proteomics, is it a genomics. In some countries interpret this data flow in a different way. They say you can create this data set, but it has to stay in the country it was collected. So the data could be exchanged, but the samples could be not sent, for example. In some countries went even so far that they said if the project includes any key which is allowing for identification of a patient, even if it's in the hands of the people who are building up the biobank, okay, it's not public key, then this data set is not anonymous. This is open. So you have to destroy everything, the data and the samples, after the project has been published and closed. Even that. So, but speaking about the scientific challenge, is simply important to think in advance, in advance. So you have to think what could be done with all these samples. For example, if I have some samples which have been used for metabolomics, and I know that there were some freeze and thaw cycles, can I use them for any other omics? For example, for proteomics and identifying peptides, it would work. Okay, not short-lived peptides like interleukins, but what peptides are there at all? It will work. If you are using urine, for example, there is as well genomic information there. There is a lot of DNA there. So it could be that usually the scientific board of revision for the ethics committee will ask you, what are you going to do with the urine? Are you going to use it for genomics or epigenetic studies? You have to think in advance what will be done with the samples and implement this already in the stage of sample collection. Because going back to patients after maybe a year, 2 years, and then you think you can use the sample for something different, it's doable, but you usually have very low compliance.
Well, thank you so much. I think that was a great way to wrap up our podcast for today. The take-home message here is as much planning as you can do beforehand to account for what it is that you're planning on analyzing, the matrix that you're using, and the patients that it's going to be affecting, the better your overall results in your sample design will be. I want to thank our 4 panelists for joining us today. You've provided some really great insight. I know that this has been helpful to our listeners. And I do want to be mindful that we mention our next podcast and to be on the lookout for any information for the overall podcast series from the Education Training Committee of the Metabolomics Society. Thank you all for joining today, and I hope you have a great rest of your day.
Thank you.
Thank you.